Okay, so now it's time to move forward and talk about other aspects of animal evolution. The skulls that you see on the screen here are here because they sort of summarize one of the steps in vertebrate evolution from the early reptiles. These three basic skull drawings represent the three basic skull patterns we see in terrestrial vertebrates. The first one here on this side, this is the skull of a reptile. This is a nasal fenestra or opening where air is going to come into the skull and then go down through to the respiratory organ. This is the orbit for the eye. And these two openings right here are for ear and muscle attachment. So these types of skulls are called diapsid skulls. Diapsid skulls are found in dinosaur and bird species. The extra two holes here is the reason for the name diapsid. For the second one here, we have the skull of a reptile mammal, general reptile mammal. There is still the nasal opening, or where the nostril would be. There is the orbit for the eye, but there's only one opening for the ear. There is no other anterior or superior opening above the ear opening in the skull. You see this in mammals and reptiles like lizards. Really primitive reptiles like turtles, which were one of the first prototype reptiles, they're like this skull here. They have, like a box turtle, has a nasal opening and an orbit for the eye, but no opening for an ear. They don't hear. Snakes don't either. There's no they feel vibrations, but they don't listen to auditory stuff, in the air. So now moving down, we need to talk just briefly about how do species, there's so many different species on Earth. Look, there are just so many birds, for example, out there flying around. How do they keep themselves all separate? You should have learned from Hardy Weinberg that, if organism populations meet each other and they're able to interbreed, they're going to mix their genes together and then they're technically one species. The question is, how do different species of frogs or different species of finches? There are 3 species of finches, for example, in North America, and they all run into each other all the time. There are goldfinches, purple finches, and house finches. While goldfinches are bright yellow or gold in color, house finches and purple finches look almost identical. To a human, house finches and purple finches can be hard to tell apart. How do the species keep each other apart? More importantly, how do they maintain separate gene pools? Well, there has been evolutionary pressure for this. If you are a member of a species and you accidentally breed with a member of a different species, whatever genes that cause that, they'll probably die with you. That's because your offspring will probably not be well adapted to where they're living. And the reason for that is they receive chromosomes from 2 different adapted sets. As a result, in their body, they have a mixture of adaptations that are probably not the best for any specific habitat. They're probably going to get eliminated. So what are the things that keep species from picking each other as mates? So when frogs get to a pond, it's dark. How do they keep each other apart? Well, these are the primary differences that species use to keep themselves apart. First off, even though frogs may all use the same pond, they may use different regions of the pond to avoid making mistakes and choosing mates that are not of their own species. So habitat differences, where two species are in the same area, but they're not in the exact same spot, and therefore don't pick each other as mates, and therefore they won't mix genes. The only way to mix genes, by the way, as an animal with another animal is through reproduction. And so using different parts of an area means that you're restricting your choices, your mate choices. Another one is temporal choices. The timing. Some frogs go to ponds early in the spring, some go later in the spring, and therefore they don't run into each other and they don't make mistakes in mate selection. There's also mechanical differences. Mechanical differences are where some frogs are big, like bullfrogs. Other frogs are small, like spring peepers. Spring peepers they're the chihuahua size, or they're tiny. They're just tiny little frogs that are as small as the flesh under your thumbnail, they can't mate with bullfrogs. They're absolutely too small for that. Mechanical differences then keep some organisms from mating with other organisms that are similar to them but not of the same species. And lastly, behaviors. Animals do all kinds of courtship rituals to keep each other apart from other species. So the birds that you see down in these pictures here, the penguins, the emperor penguins, and the crested tern courtship behaviors are what species use to determine, are you one of my group? Because most of the time, species do not want to mate with members that are not in their group because their chromosomes will not survive in their offspring. This entire set here of evolutionary forces that keep species apart are called prezygotic isolating mechanisms. They're prezygotic, meaning they occur before species actually reproduce, and they keep species from reproducing with each other. They're very effective. So that's the reason why we have so many different species of birds and frogs and insects in North America. They keep themselves apart with all of these things by not breeding in the same place, not breeding at the same times, having mechanical differences that disallow specific reproduction, and courtship rituals that keep themselves apart. The last thing, it's a little rarer, gametic isolation. What this is the animals don't keep themselves apart. They actually, the two animals actually mate with each other that really shouldn't be mating with each other, yet the sperm from the one species cannot successfully fuse with the egg of the other species, either because the egg won't allow it or the sperm has something on it that prevents fusion. As a result, even though the behavior, the mechanical differences, temporal, and habitat failed, the gametes fail to fuse, and that keeps the species safely apart and the offspring from being a complete waste of time. Do you understand that anytime interspecific reproduction occurs, it's often a complete waste of time and effort on the part of the parents because the babies end up all dying. 100% death is not unusual when that happens. Now, there are a few post-zygotic isolating mechanisms that do occur. These are mechanisms that occur between species that are different. The species mated in the same place. They didn't successfully keep themselves apart with any courtship rituals or gametic isolation, but still their chromosomes don't mingle because ultimately either their babies, which are now hybrids, they're either inviable, so the babies start to grow up and then they die in the womb, or the hybrids do grow up, but they're sterile. This does happen with donkeys and horses. The offspring of the donkey-horse courtship and reproduction is the mule. Mules themselves are hybrids that cannot reproduce. Therefore, donkeys and horses are separate species, even though they will mate with each other. In a pasture, hybrid sterility keeps the two chromosome sets apart and prevents them from merging into one species. And then we have hybrid breakdown, which is rarer, but this is where two separate organisms, two separate species mate. Nothing could keep that from happening. The hybrids were not inviable, they were not sterile, but the hybrids then go on to reproduce. But even though the hybrids are fertile, the next generation of offspring are infertile and they fail. Now that's rare, but that does happen in a few species. In a lot of plants, you see hybrid breakdown, where second and third generation plants from the hybrids fail to successfully reproduce. I don't know of any examples in animals, but it does happen in plants. Now, since we're looking at patterns of evolution, just how common is just in general evolution? Well, the first most common pattern of evolution on Earth is allopatric evolution. Allopatric speciation is the evolution of new species in different areas. Allopatric speciation is really common with species that get spread out into large geographic regions, and then the species evolve differently in the different regions. This kind of evolution is said to be divergent. What that means is you have two species coming from a single ancestor, and they're evolving away from each other. Basically, allopatric speciation is caused by genetic isolation. Gene pool isolation, meaning one set of genes is separated from another set of genes, and as a result, natural selection grinds on the two populations separately, and you end up with two separate species that end up being separate from one another because of these differences being prezygotic or the other differences being postzygotic. So ultimately, prezygotic and postzygotic mechanisms will separate allopatrically speciating animals. Most recently, we've had Pleistocene glaciations here in North America were a big deal. That gave rise to all kinds of different frogs and birds in North America. And we see that across the continent. Generally, allopatric speciation is pretty slow. It's only going to happen across millions of years. It happened on the Galapagos Islands. It happened in Hawaii. The founder effect makes it a little faster because genetic drift can happen in those new areas. But ultimately, the isolated populations evolve rather slowly. So that brings us up, though, to a different kind of speciation, which is a lot faster, and that's sympatric speciation. Sympatric speciation occurs when a group of plants or animals living in an area have a freak reproductive event, and as a result, their offspring are not really a member of the group, but they are viable and they can reproduce amongst themselves. That's most commonly experienced as polyploidy. Polyploidy is where usually two sperm unite with one egg and you get a triploid offspring. That happens a lot in plants. The potato, oats, tobacco, and cotton are all polyploid. So are bananas. Polyploid plants tend to be real vigorous growers because they have extra chromosomes. Polyploid plants are common because they are not killed by embryonic development, but polyploidy in animals is truly rare because polyploidy in animals usually kills animals during embryonic development. So sympatric speciation is pretty rare, but it tends to be very rapid when it happens. Allopolyploidy is where chromosome sets of two different species do the uniting and forming the triploid or more zygote. Autopolyploidy is where two or more chromosome sets from the same species form the zygote. This allowed plants, sympatric speciation is actually pretty common in plants, and a lot of our crop plants are polyploid plants produced by sympatric speciation. It's not an accident that they're crop plants, sympatric speciated plants, because they're almost always polyploid, they tend to grow more vigorously than their diploid counterparts, and they end up on the farm because they can make food more readily than their diploid counterparts. Most animals are diploid. A lot of plants are diploid, but many plants, especially those in agriculture, are polyploid because they're fast growers. So that concludes this aspect of patterns of evolution.